members is just 12, and he is here facing up all of us to learn public speaking. He is going to be talking about life of a cell, Alan.
What is the objectives of this speech?
The objectives of the speech, he's giving his third project, and he is to have get to the point is the main theme, where he has to have a general and a specific topic in his speech, and it should have a good beginning, body, and conclusion. And that's about it. So welcome, Alan.
Did you know that there are over 1 trillion cells in our body? How much do you think there would be in an elephant? In a gigantic blue whale? Now, the history of cells started when the scientist Antony van Leeuwenhoek looked at a sample of pond water under his powerful microscope and found cells. The life cycle of a cell has 3 phases: the interphase, mitosis, and cytokinesis. During interphase, there are 3 subphases: G1, S, or G2. G1 stands for gap 1, S stands for synthesis, and G2 stands for gap 2. In G1, which is where the cell spends in interphase, the cell spends most of its life. If you would count the life cycle of a cell to be exactly 1 day, the cell would spend 23 hours in interphase. In gap 1S and gap 2, the cell grows and develops. Only in the S phase, the cell will start to duplicate its DNA, and later they will wrap together to be chromosomes. And this is a cell during interphase. As cell division, which is how cells reproduce, the cells will make an exact copy of their DNA, then split together, and they will grow, and each daughter cell will reproduce again. This is a typical diagram of a cell. These are organelles, or tiny organs. Each cell has functions like a mitochondria, which helps the cell produce energy, or chloroplasts, which transform sunlight energy, and they are green, and that's what makes plants green, and they are only found in plant cells. This is a typical animal cell. An animal cell is round, and an animal cell does not have an enormous central vacuole. A plant cell, however, is rectangular and has a gigantic central vacuole. A vacuole is a big storage container that is about 4 times the size of its nucleus. So now the DNA is copied, and now we enter the second phase of cell division, mitosis. In mitosis, most of the action happens. The first subphase of mitosis is prophase, and that is when the DNA coil up together to become chromosomes, and then the chromosome is now visible. At the end of prophase, the nuclear membrane around the nucleus starts to disappear. So this is a cell during prophase. As you can see, these H-shaped DNA are coiled together. This is a DNA, these are 2 DNA and coiled together by these. But a real coiled together DNA looks somewhat like 2 rubber bands linked together. The next phase is metaphase, and that is when spindle fibers grow from the ends of the cell and align the chromosomes in the middle. This in the metaphase looks like a gigantic watermelon. However, it is not gigantic. In this watermelon, you can see that these 4 chromosomes are lined up at the center by the spindle fibers at the ends. The spindle fibers grab hold of the ends of the rubber bands, In the third phase, anaphase, this is where the action really starts. So the spindle fibers then pull the chromosomes, and because there is a spindle fiber on each side of the chromosomes will come apart. So once the spindle fibers pull hard enough, the chromosomes will split, resulting them to come together if they pull hard enough. In anaphase, this is what a cell would look like. And as you noticed, and I can show you a normal cell as interface. The anaphase cell is actually quite much longer than this interphase cell. And that is because the spindle fibers push the chromosomes this way as they start to split. Now during anaphase, all the other organelles, they also do the same exact thing as what the cell does, and they make a copy of themselves as well. So now there's two nucleuses, two mitochondria, two vacuoles floating around in the cytoplasm, and that is what holds the cell in place. The last subphase of mitosis is telophase, and that is when the cell starts to split. This is what a telophase cell would look like. In telophase, the chromosomes go to each end of a cell, and the nucleus reforms around the chromosomes. And the chromosomes, they are no longer visible. Now, onto the third and final phase of the cell, cytokinesis. In cytokinesis, there is no subphases. Cytokinesis does not last very, very long. Cytokinesis is the final process in where the cell splits. In cytokinesis, the nuclear membrane forms again. Everything is back in place, and the cell is ready for another division. Now, one of the subphases that I have not mentioned in this cell cycle is G0, or gap 0. And in this phase, the cell doesn't divide, it doesn't do anything. And now you know all about cells.
Thanks, Anna. We need to say thank you, Professor Anna. So,